Hello friends, I'm Shubham and today I'm going to talk about ABO blood groups. Actually, as you all know, there are four major kinds of blood groups, that is blood group A, B, AB and O blood group. Actually, the naming of these blood groups are depend on the types of antigens they have. For example, if we talk about the blood group A, if we talk about blood group A, by the way, before talking about the blood group A, what I'm going to talk about in this video, that I will talk about the antigens, which are also called as agglutinogens, because these antigens are responsible for the clumping in the blood transfusion reactions if there is any incompatibility with the blood groups. That's why these antigens are also called as agglutinogens. So we will talk about the antigens, we will talk about the antibodies of the blood groups, and then we will talk about the compatibility and incompatibility of the blood groups, and then in last we will talk about the blood transfusion reactions, that what could be the severe reactions while transfusing the blood. So let's come back and talk about the blood group A. So if we talk about the blood group A, blood group A have A antigen. Blood group A have A antigen. Along with that, blood group A have anti-B antibodies. Blood group A have anti-B antibodies. We will discuss about these antigens antibodies, that why blood group A have A antigens and why this blood group have anti-B antibodies, not anti-A antibodies or any other antibodies. Why is it so? We'll talk about in this lecture later on. But before that, let me summarize the blood groups, that blood group A have A antigen and anti-B antibodies. Like that, if we talk about B blood group have B antigen. B blood group have B antigen and anti-A antibody. Anti-A antibody. Blood group B have B antigen, that's why this blood group is named as B. If any blood group, for example in blood group A, it have A antigen, that's why it's named as A blood group. Now likewise, if we talk about the blood group AB, what about blood group AB? This blood group AB have both the antigens, that's why it is named as blood group AB. It have A antigen, this will have A antigen, as well as this will have B antigen at the same time. Now as this blood group AB have both the antigens A and B, that's why this blood group AB will not have any antibody, will not have no antibody, no antibody. Now if we talk about blood group O, blood group O does not have any antigen, that's why this blood group is named as blood group O. So here we have blood group O, as blood group O don't have any antibody, don't have any antigen, no antigen, don't have any antigen, that's why this blood group is named as O, which means no antigens. Now if we talk about the antibody, blood group O will have both the antibodies in their blood, that means it will have anti-A antibody as well as it will have anti-B antibody at the same time. So it will have anti-A antibody and anti-B antibody. Now, these were the basic classifications of this blood group, that what kind of antigens and antibodies do they have. Now, let's go into some details of these blood groups and know about some details about the antigens and antibodies. So, please allow me to rub this stuff. Now, as I told you that blood group O don't have any antigen, but they do have something on their membranes. Their RBC membranes do have something. Now, what is that? Actually, blood group O, if we talk about the cell, of the blood group O, for example, this is the cell of red blood cell. This is the premature RBC while we are developing our RBCs. So there are reticulocytes, and as we all know, the reticulocytes will further go into the process, and there will be the mature RBC. So I am talking about the premature RBC, which those RBCs, they have the nucleus. However, a mature RBC don't have nucleus. So if we talk about the nucleus over here, okay. Now, for example, these are the chromosomes over here. One is paternal and one is maternal. Now, at one locus, for example, now they don't have any antigen, okay, so they are not producing, like further I will talk about the antigen and antibody, what they have in some special molecules. But what is the significance of this blood group O? What is the specialty of this blood group O? Actually, they have one gene, one chromosome over here, and one gene of this chromosome will produce one special kind of enzyme. Now, what is that enzyme? Actually, that enzyme is fucose transferase. Now, this enzyme will transfer this fucose onto the membrane of this RBC. Okay. So this fucose will bind with these are the oligosaccharides, the chain of monosaccharides, and this whole group of fucose along with the monosaccharide chain, this whole group, this is antigen O. This is antigen O. Actually, we have one special kind of substance in this blood group O. This is H antigen. H antigen. However, we call it antigen O, we should call it antigen H. Because as we know, they don't have antigen A antibody, antigen A or antigen B, but they do have H antigen. Now, let me talk about the other blood groups. What about blood group A, blood group B, and blood group AB? Blood group A, B, and AB, they will have the same substance, H antigen, but and this chromosome was the chromosome number 9. Chromosome 9. Okay. So let me talk about the other blood groups. For example, let me take the example of blood group A. Along we took the RBC, premature RBC, and here we have the, for example, the nucleus, and these are the paternal and maternal chromosomes. Okay, again, but in this case, along with the fucose transferase, there is one more specific chromosome, that is chromosome number 19. Chromosome 19. So what it will do? For example, here we have chromosome 19. Chromosome 19 will have the special enzyme. Like here, we had the special enzyme fucose transferase. In case of blood group A, we have the special enzyme which named as transferase A. So, transferase A enzyme This transferase A enzyme will send the special molecule, that is NAGA, N-acetyl galactosamine. N-acetyl galactosamine along with this substance, which is H substance, Now, this whole group we called as actually antigen A. If we talk about the blood group B, what they have? If we talk about the blood group B, for example here, same we have RBC, we have the nucleus inside it, we will have the paternal and maternal chromosomes, paternal from the father, maternal from the mother, and this time also we have the special chromosome, that is chromosome 19 again, as in like in the case of antigen A, blood group A. So this is about the blood group A. This was about the blood group O, and this is about the blood group B. Okay, and here we will talk about the blood group AB. Now, this special chromosome 19, will produce a special enzyme, that is transferase B. With the help of transferase B, it will attaches the special molecule, that is galactose along with H substance. So this combination we called as antigen B. Okay. Actually, the S substance is very common. H substance we have in blood group A. S substance we have in blood group in blood group O, we have in blood group A, and we have in blood group B. And S substance we have in blood group AB as well. So again, if we talk about blood group AB, we have this cell, we have the nucleus, we have the chromosomes, paternal as well as maternal. We have the enzymes. We have the special chromosomes. In this case, like in blood group A, we had the chromosome 19, and in blood group B also chromosome 19. So here also we have the the chromosome 19, and as well as chromosome 9, because they also produce the fucose. So chromosome 19. Along with the both the enzymes, blood group AB, there are both the enzymes transferase A like in blood group A and transferase B like in blood group B. So there are both the enzymes in blood group AB. So with the help of both the enzymes, they will present transferase A as well as transferase B. With the help of both these enzymes, they will attach here NAGA as well as galactose to the H antigen, and this whole group we will call as antigen AB. Actually, at some places NAGA can attach to the H substance. At some places galactose can attach to the H substance or H antigen. So in the blood group AB, there will be both the enzymes transferase A as well as transferase B, and which can represent both NAGA and galactose with the H substance, and will produce the antigen AB. Now, further, let me talk about the genotypes of these blood groups. Let me classify this. Let me talk about the genotypes of these blood groups. So this blood group A could have two genotypes. For example, over here, those maternal as well as paternal chromosome can take part in the blood grouping. So it would be IA genotype, because both the chromosomes from the mother and as well as father, they both are participating. Now, for example, if one is participating, for example, doesn't matter if it is maternal or is it paternal. So one of the chromosome is participating and the other one is not participating. So we could have another genotype, that is IA and IO. In both the cases, the person would have the same blood group, that is blood group A. For example, in this case, so blood group A will have two genotypes. Blood group A will have two genotypes. Now, if we talk about the blood group B, the blood group B also have, for example, one maternal chromosome is participating in the blood grouping, and another one is not participating. For example, paternal one is participating and maternal one is not participating. So we could have the genotype IB and IO. And if both the chromosomes are participating in the blood grouping, so we could have the genotype IB. So here also, like in blood group A, we have two genotypes. Now, if we talk about the blood group AB, the blood group AB have both the antigens, as I told you earlier, that blood group AB have antigen A as well as antigen B. So they will show the codominance. That means IA and IB, both these maternal and paternal chromosomes will take part in the blood grouping system. So they will have only one genotype, that is IA-IB, and that's why this shows codominance nature. These are codominant. Okay. And if we talk about the blood group O, then we have no antigen, so there will be IO, because no one is participating in blood grouping because they don't have any antigen. So that would be only one genotype. So these were about the genotypes of blood groups. Now, why the blood group A have A antigen? If the blood group have A antigen, so why they have B antibody. For example, in blood group A, they have the antigen A, but why they don't have anti-A antibody? They have the anti-B antibody. Why? Because in earlier life, before birth, we don't have any antibodies actually. After birth, we started to develop the antibodies. So, if a person have blood group A, that person will have the antigen A along with him. So, our immune system will start to presenting these antigens to our immune system, and those lymphoid cells who reacts to this antigen will be destroyed, and you can say will be suppressed by the immune system. So that's why if a person have, for example, blood group A, that will have antigen A, so they don't produce antibodies against its antigen. So that's why the person with the blood group A with antigen A will not have anti-A antibodies, rather will have anti-B antibodies. Now, actually, after birth, when we start to take our food, so with the food we ingest bacteria also, and in those bacteria we can come across with the various antigens. For example, if I have blood group A, I will have antigen A, but if any other antigen will come into my blood, so what will happen? My body will start making the anti-B antibodies and will try to get rid of this antigen. So that is the reason behind that why any particular blood group have the same antigen and opposite antibody. Now, if we talk about the compatibility and incompatibility of the blood groups. Compatibility and incompatibility of blood groups. That what blood group can be given to which patient. For example, blood group A can be given to which blood groups, and blood group B can be given to which blood group. For example, if we talk about the blood group A can be given to blood group A as well as blood group AB, because as the blood group A have the anti-B antibodies, so they will not make, ideally they will not make the antibodies against A antigen, because this blood group will have A antigen. And same in blood group AB, they will not have any antibodies, so they will not react to the antigen of A. So that's why the blood group AB is called as universal recipient. So this is the universal recipient. Okay. Now if we talk about the blood group B, again it can be given to blood group B person and blood group AB person. AB because they don't have antibodies again, and B will have the anti-A antibodies, so they will not react to the blood group B. Now if we talk about blood group AB, this blood group can be given to the AB only. Why? Because they have both the antigens. If we give it to the A, then anti-B antibody will react to this. If we give to B, then anti-A antibody will react to this. So, and if we will give to the O, they have both antibodies, anti-A and anti-B. So they will definitely react with this blood group. So AB blood group can be given to the AB blood group persons only. Now if we talk about blood group O blood group can be given to the blood group O as well as other blood groups, blood group A, blood group B, and blood group AB as well, because they don't have any antigen along with it. So they don't have any antigen, so the antibodies of these different kinds of blood groups will not be able to react to this antigen, react to this blood group. So blood group O is also known as universal donor, also known as universal donor. Now one more thing, that these antibodies are known as agglutinins, because as I told you earlier that antigens are responsible for the agglutination reactions. That's why they are called as agglutinogens, and as they bind with the antibodies, that's why the antibodies are called as agglutinins. Actually, the donors, when in the blood transfusion reactions, the donors RBCs are destroyed by the antibodies of the recipient blood group. So the recipient RBCs are not destroyed actually. Actually, the RBCs which are destroyed in the case of incompatibility, those are donors RBCs. Okay. Now let's have some talk about the blood transfusion reactions. The antibodies involved in this blood grouping system are usually immunoglobulin M and immunoglobulin G. Immunoglobulin M, this is actually the pentameric molecule. It is actually like this. It is actually like this, and they could capture along with them at least 2 antibodies. For example, over here. So they can bind with 10 antibodies along with them. So here we have, for example, here RBC, for example, in case of any incompatibility, for example, we give the blood group A to the person who have the blood group B. So in case of clumping, these antibodies, for example IgM, they could capture the 5 RBCs and, for example, 10 agglutinins or 10 antigens along with them. So this is the molecule of IgM. So actually they have each antibody have 2 binding sites along with them, and this is the pentameric molecule. So this was the discussion about the ABU blood group. In the next section I will talk about the RH grouping system. Till then, take care, see you soon.